Thanks, Nina. Good morning, everyone. So my presentation today is a sort of a review on EMG detection systems. And this is because you have, many of you have been in all our workshops and you have learned a lot about what we have developed. And many of you have only worked with high-density EMG, with decomposition of high-density EMG. And you may believe that this is the standard. But actually, this is a development of the last 20 years, approximately. There was really an hype on this topic, but this is not how everything started. So I think it's also important, especially for students, to know a little bit about everything, how everything started. So I will talk a little bit, as I say, I will provide a sort of historical perspective, both of intramuscular and surface EMG, and the latest development of multi-channel EMG, and especially flexible electrodes, which is an active field of research now. So you all know that the muscle is made by fibers, and these fibers are controlled by motor neuron at the spinal cord. So each motor neuron controls a set of fiber, and together they form a motor unit. And you also know that basically the motor neuron integrates the inputs, and when the potential is above a certain threshold, it discharges an action potential that travels to the muscle fibers through the axon, and then it propagates towards the two tendons. So if we place an electrode on top of the muscle, we will record what is called a motor unit action potential, which is the sum of the contribution of the action potentials of individual fibers. And it's because the neuromuscular junction is very safe. We have a one-to-one correspondence between motor neuron action potential and motor unit action potential. So but how were the first action potentials recorded in humans? So the term electromyography dates back to 1980 and was proposed by Marais, who is also very famous for studies in kinematics. You may have, if you work on the movement field, you have heard about him for sure. But at that time, actually, he didn't record electrical signals. He used a mechanical device, but still to show the activity produced by muscle. The first recording of single motor unit action potential comes from Adrian and Bronk in 1929. And those are the researchers who introduced the concentric needle EMG electrode. So this electrode, you have an outer cannula that works as a reference, and then you have an inner core in metal, that is depicted in black, and this is actually the active electrode. So, and thanks to this measure, Adrian and Bronk were also the first who were able to see the association between electrical activity in the nerve fiber and in the muscle fibers, which is the rationale behind all this processing that we do nowadays in signal from the muscles. We can do that basically to infer the spinal activity because of this association that they observe in this study that was done almost a century ago. And this study was very important because it's the one who started the development of in vivo motor unit physiology. So this technique is still used nowadays. We have a century of history where this technique has been used. And in about 20 years later, a new needle was proposed, the monopolar needle. And this is somehow similar, but here, like the active part is the tip of the needle. And here you have a sort of summary provided by Gao and colleagues, where they actually show how the field of needle EMG evolve over time. So the first electrode that was proposed was the concentric needle. And concentric needle can actually exist in different variants. You can have only one metal as the core or two, and have a so-called concentric bipolar. You can have small electrodes in case you need to target small muscles as those of the face. And then you have the monopolar needle, where instead the active surface is the tip. And you can see here on the right side that depending on the needle you use, you detect motor units from a certain area that is a little bit different depending on the electrode that you use. But what you can certainly observe here is that these electrodes are very selective. So you see, like, this pink area is the detection area. So if motor units are within the pink circle, the electrode will be able to see them. And if they're outside, it will not. And then, another development by Stolberg was the single fiber electrode. In this electrode, you have, again, a cannula and a core of metal, but this metal is very, very thin, and it gets in contact with the muscle in this side port of the cannula. And because it's really thin, it's actually able to detect the activity of only 1 or 2 fibers within a motor unit. So when you use monopolar needles or concentric needles, what you get is the activity of 1, 2, 3 units. And this, you also get the activity of many fibers belonging to the same unit. While in this case, you have only 1 fiber. And this electrode has been used a lot, like, for instance, for studies in conduction velocity, and many of the findings are still very valuable nowadays. And this electrode can also exist in a slightly different version. This is the so-called macro EMG electrode. So you have the same wire on a side port. So this is a single fiber electrode, but you can also calculate the potential difference between the cannula and the reference. And in this case, what you get is an electrode that is much less selective. So you see here that the detection area is much bigger. And in this case, the idea of the rationale why this electrode was built was to detect the signal from all muscle fibers belonging to the same unit. Now, what is amazing about these systems is that, after 1 century, they are still used nowadays. They are used in research, but also these are the only EMG systems that are used in clinical settings. And the limitation is that, of course, they are invasive, even if minimal invasive. With the invasiveness comes some discomfort. Some people are afraid of needles. Some people may not have needle insertion because of some diseases. And they are also stiff. This is metal, and stiffness, of course, implies a little bit of discomfort, but also implies, like, if you have any movement, then the action potential shape will change, which makes the identification of action potentials more difficult. And at least to solve the problem of stiffness, this is why fine wires were introduced. And we have some studies in the 50s, and then Basmesian and Stecco actually produced a paper where they explained the technique so that researchers could actually build these electrodes in the lab. And how does it work? Basically, you have an hypodermic needle. And what you do, you thread 2 wires inside. So these wires have a metal core, and it could be steel, for instance, and then they are covered by some sort of insulator. It could be Teflon, for instance. And you insert 2 so that you can have a bipolar detection. And then at the tip, you remove part of the insulation. So you see here that you have bare metal, this part in gray. And depending on how much Teflon you remove, this detection will be more or less selective, okay? So differently from the electrodes that I showed you before, where actually the needle is used to detect the electrical signal from the muscle, in this case, the needle is used as an intruder. So you insert the wires into the muscle with the needle, but then you remove the needle and only the wires stay inside for the recording. So the advantage compared to the needle electrode is that these electrodes are, the wires are less stiff, and therefore the level of discomfort is a little bit less. So of course, you still have the pricking sensation from the insertion because you need to put them inside the muscle. And the disadvantage is that what you have said before, what we have seen before for the needle electrode, they are still invasive, even if minimal invasive. And normally, when you use a needle electrode for EMG recording, you can tailor a little bit the position in order to get, for instance, a motor unit with a very high amplitude action potentials. While with the wires, once you have insert them and you remove the needle, you cannot reposition them. So you record what you can get in the position that the electrodes are. And sometimes this position is good, sometimes is not ideal. And this is why people have proposed different electrodes to take this into account. One very famous is the quadrifilar proposed by De Luca. And here the idea, this is still a needle electrode, it's not a wire, but you have 4 wires within the cannula of a needle. And in this way, you can record 4 monopolar signals. So you can see which one is better, easier to analyze. Or you can also make different combinations and calculate 6 different bipolar electrodes. So that basically you can choose the one that is most selective, so the one that is easy to extract motor unit action potentials from. Or you can have also electrodes where you have, a wire that is insulated, as you have seen in the fine wires, and then you occasionally remove some of the insulation, so that also in this case you can do you can expose a very, very little area in order to get electrodes that are very selective. So selective that you can also record the signal when the force levels are high, but not selective enough to record single muscle fibers action potentials, as is possible to do with the needle. So we have, I have repeated quite some time that intramuscular EMG is selective. And what does it actually mean? So you can observe here a trace of 10 seconds, I believe. And you can see that you have a part of the signal where there is just background noise, and then some spikes that stand out from the baseline. And this is because, EMG are so selective that needles and wires are usually so selective unless you use macro EMG. That you can detect the activity of individual units. And these normally have a little degree of overlap, at least a low force level. So because when you insert your wire, so in this cartoon, you have a muscle cross section, and different circles are different motor units. So since this electrode has only a very small exposed area of the metal, what you'll get is the activity of 3-4 motor units. Originally, actually, these signals were sorted by visual inspection, or like you make the electrode so selective that you detect a unit, only 1-2 units, and you simply threshold the signal. But then in the 70s, De Luca started introducing like the first decomposition algorithms that are those algorithms that actually process the signal in a semi-automatic fashion, so that basically a lot of computation actually is not done by the operator. And specifically, like these signals from invasive electrodes are normally sorted via template matching techniques. So for instance, in this signal that you have here, you can actually recognize 4 different action potentials belonging to different motor units. And since the similar is flow shape and you have a little degree of overlap, you can actually find out when the signals that stand out of the baseline belongs to one of these 4 motor units. So summing up about intramuscular EMG, like as I mentioned, this is still the standard in clinical settings. And also for 1 decade has been consistently used for research on single motor unit physiology. So recruitment, discharge rate, coherence, and so on. But it comes with limitations that we have mentioned a couple of times. The fact that it is invasive, is uncomfortable, and but this is mainly from a subject point of view, but also from a signal processing point of view. Since these electrodes are very selective, what we get is actually a limited view on the motor units that made up a muscle. And this is actually the reason why high-density surface EMG or decomposition of high-density surface EMG was introduced. So, you know, a muscle the muscle with the smallest number of motor units have hundreds of motor units, but we can have muscle with many more than that. And when we record through 2-3 motor units with invasive EMG, an obvious question is how representative is this of the entire muscle? And this is why people have tried to circumvent this limitation using other technologies. So surface EMG originally was mainly recorded with bipolar electrodes. So you place 2 electrodes normally on the same side of the innervation zone, and you record a signal like those that are the compound activity of many motor units. And these electrodes are used mainly to understand the activity of the muscle. So if you calculate the envelope and then you extract the amplitude, higher amplitude is a sort of proxy of muscle activation intensity with many limitations. We know that amplitude depends on the thickness of the fat layer, depends on the characteristic of the electrodes, but under the same condition, you can have a relative comparison. And you can also understand timing of activation. So when a muscle becomes active, the amplitude will increase compared to the baseline. But it's also used, surface EMG is also used for studies on biofeedback and also in the rehabilitation setting. So for instance, like in prosthesis control, since the 50s, actually when the first myoelectric prosthesis came out, surface EMG signals recorded from the muscles are actually used in order to infer the user intent and control a prosthesis depending on this decoding. But it is in the 80s, people started to record the surface EMG with multiple electrodes that were originally placed in a linear configuration. So you have a so-called monodimensional electrode. The first studies were from Masuda. When you record the signal with arrays, you can actually follow the propagation of an action potential from the innervation zone, where the signal changes polarity, to the two extremes, so like towards the tendon of the muscle. And this electrode is specifically used to calculate the innervation zone, as I say, where the signal changes polarity, to calculate the fiber length. You can observe here that here you have an action potential that propagates, but then when it reaches the tendon, you have this non-propagating component. So in this case, the information you can infer is that the fiber length goes from here to here. And this electrode is also used to determine the optimal electrode positions, which is normally placing the two electrodes, like a pair of bipolar electrodes on the same side of the innervation zone. And there was a full European project aim to standardize the electrode position, the SINIAM project that you are probably very familiar with. But there are also most recent recommendations that you can find in the tutorials published by the Journal of Electromyography and Kinesiology, and also the outcome of the SEED project. Now, immediately after, actually a few years later, the introduction of the monodimensional arrays, a Dutch group led by Segeman proposed the use of bidimensional electrode grids. So this electrode actually allows us to calculate heat map, so to understand which region of the muscles are more active. But more recently, they have been used actually to enable decomposition of surface EMG into motor unit action potentials. And these were the very first studies by Stegeman, by Oloban, and nowadays there are more groups working on this topic. So why do we need many electrodes in order to, many surface electrodes in order to decompose the activity of individual motor units? Like if you look to a EMG signal, a surface EMG signal, so you see that compared to the selective electrode that we have seen before, this surface EMG is much less selective. So you have the activity of many motor units overlapping together. So by naked eye, you cannot see what is actually embedded in this signal. And the algorithm cannot do either. But then the idea was that if you actually place many electrodes together, you may be able to identify the signature of individual motor units. And you have seen Alex this morning presented this a study or like the idea of the blind source separation. So you have many people talking and you have microphone listening to them. And there are signal processing techniques based on blind source separation that enable to distinguish the sources based on the signal recorded at the microphone. Now, in our case, we don't have people talking, but we have motor units firing. If we place the electrodes, we record the contribution from different units, and we can use the same family of algorithm in order to identify the activity of single motor units. And these algorithms are called blind because they don't have many assumptions. But there is a very strong assumption, which consists in the fact that the number of electrodes that you use has to be at least equal, better if greater than the number of sources, which is the number of motor units. So we have said before that in one muscle, you have 100 of units. Of course, there are not all active at low force levels. But if you want to use this class of algorithms, you need to have many electrodes in order for this hypothesis to be fulfilled. And so what we have said a lot during this series of workshops about the decomposition of high density EMG. So I will just summarize in a slide that being non-invasive, they can actually be used largely, so also in patients, in children. And compared to invasive EMG, you can detect a much higher number of motor units. But the point is that this was very well explained in the Matei talk, like they can only be used, surface EMG can only be used to record from superficial muscles. And the view that they provide from the motor units activity is very biased in the sense that only those units that are either close to the surface or that are very big, so the action potential has a large amplitude at the surface, can be detected. And this is why we actually work on the development of high-density intramuscular EMG. And the idea that we had was, wire EMG are very selective. Each wire can record the activity of few units. But if in one wire we place multiple detection systems that are also very selective, we'll be able to record the activity of multiple units. And this is our first study where we had a multi-channel electrode with 16 detection points. These detection points are very small, but they are far apart, 1 millimeter from each other, so they can pick up the activity of different units. And this electrode, like the technology or the insertion technology, is very similar to the classic EMG. So you have a needle that is used as an intruder, and the wire in the old-fashioned electrode was inside, like the wire was here. But in this case, we keep it outside because there is a big connector. We need to accommodate 16 electrodes. So of course we cannot remove this from the app. And now, if we use an electrode like this, and we tailor the distance between the electrodes depending on the target muscle, we can actually perform a spatial sampling of the electrical activity across the whole cross-section. And here you have signals recorded from the abductor digiti minimi, and you can see with different colors, like we have different motor units. And sometimes you can see the same motor units in multiple channels, but then we can actually identify those motor units that are unique. And since instead of a small portion, you actually sample the whole cross-section, in this case, you have a much better sampling of the motor unit population. And also, like, since the electrode is within the muscle, you can actually detect the activity of small motor unit as well. So your sample is less biased. Now, the problem with this is that, we can decompose these signals via template matching, as I explained at the beginning. But if you have many channels, you also need a lot of time for processing the signals. And this is why we actually thought that it would be much better if we could decompose the signal with the blind source separation methods that have been proposed to decompose high-density surface EMG. You may remember that the assumption is that we have more electrodes than motor units. So 16 electrodes are definitely not enough. But our latest prototype actually includes 40 electrodes. These are alpha millimeter far apart. So like the technique is very similar to before, but just we have a bigger connector in order to accommodate for more electrodes. And in that case, we can record a much bigger population of motor units. So this is a study that we have run with Imperial. And basically, you can see that with 2 electrodes, we can detect the simultaneous activity of 80 motor units. And the good thing is also that many of them can be automatically extracted via blind source separation. And this study was done in collaboration with Alex, and with the algorithm that he proposed for surface EMG detection, we actually had a very accurate decomposition, 99% on average, which is without any interaction from the user. And this data have been obtained by comparison with data that were manually decomposed. And this is considered the gold standard in intramuscular electromyography, especially when you have multiple channels, so you can compare the activity of the same motor unit in different electrodes. So we have applied these electrodes in many experiments, like in different institutions, like superficial muscles, but also deep muscles. This was a study done at the Neuroscience Research Australia Institute. This is a recording from the muscle that controlled the tongue. And we have also done recording in patients, specifically in tremor patients, but also a lot of animal studies, specifically in rats, in collaboration with the Medical University of Vienna, and also in non-human primates with Johns Hopkins University. And other people, and now this field is also expanding, and other groups are also working on the development of electrode. For instance, in this case, a specific electrode has been developed for animal study. So summarizing, now these electrodes, like high-density intramuscular EMG, can be used both for detecting the activity of superficial and deep units. They can also be inserted in deep muscle. You have seen the example of the genioglossus, a muscle that contributes to controlling the tongue. They are very thin, like if you compare the thickness of the fine wires and the thickness of the metal in our electrode, that's much, much thin. It's on the order of nanometer. And because of that, these electrodes are very flexible, which means that once you insert them, basically you cannot perceive because like the maximal thickness is only 20 micron. And you can get a higher number of motor units. The cons is, of course, that, again, also those are invasive. At least for inserting the electrode, you need to place the needle. So originally, this EMG was mainly used for a physiology study, really to understand the principle of motor control. But recently, there is an increased trend in using EMG, both surface EMG, intramuscular EMG, single-channel EMG, multi-channel EMG as neural interfacing. And this, as I say, has been done with both surface and intramuscular. And just from a quick search on PubMed about high-density EMG and control, now I don't have a scale here, but just this gives you an idea of the relative increase throughout the year. So now this is a very, very popular topic. And the good thing when using surface experiment with surface electrode are clearly easier to implement, but also with intramuscular electrode is not so complicated because of the modality that you use to insert electrode. So you use a needle as an intruder, then you remove. Like this allows to perform acute experiment without any need of surgery. So you can actually test algorithms simply in acute experiments. And this is a study by Agnese Grison in Imperial. So where she actually performed this very nice experiment where she introduced high-density intramuscular EMG into forearm muscles. And she also placed surface EMG on top, high-density grids. And basically, she decoded the activity during finger movements. And a result showed that the decoding with high-density intramuscular EMG is more accurate. And the reason for that is that muscles from the forearm are very small, they are very close to each other. So when you place electrode at the surface, you get a lot of crosstalk, while with the intramuscular electrode, you can actually target the specific muscles that you are interested and be more selective, but at the same time get a lot of information from the motor units that you can extract from these signals. So summarizing, I sort of present an overview of both the surface electrode and intramuscular electrodes. And a question or something you may wonder is which method is actually better. And I would say that both methods have pros and cons. Of course, like the big plus of surface electrode is that they are superficial. So like the applicability can be very widespread. But at the same time, it's not something, it's not a technology that you can use to assess deep muscle. But so the choice really depends on the application. And you have seen the trends and how technology evolves quite rapidly. And nowadays, also Alex presented this in his first slide, but there is a trend towards producing electrodes that are very thin, that are micro machine, that are so flexible that they conform very well to the skin shape. So you see here we have electrodes placed at the tip of the finger, and you can see here that there are, so flexible that you can actually fold. And when you want to trash them out, you can make them fit in a very, very small place. So I'm confident that there will be more development in the future about these electrodes that will be so flexible and so not intrusive that we'll almost not even perceive them while they are on our skin. So yeah, this is basically the slide I presented at the beginning, and basically I sort of discuss how the technology for surface and intramuscular electrodes develop over time. And hopefully we have a bright future ahead of us where we'll see many more development on this field thanks to flexible electronics. And I'm happy to take any question if you have.